I'm now going to test the same foodstuff for the presence of oils. Oils are classified as a fat, which is liquid at room temperature. But we need a slight deviation from the usual procedure. We're going to put our sample of biscuit in there. Just going to crush it a little bit. And this time, instead of distilled water, I'm going to add industrial methylated spirit. The industrial methylated spirit will liberate any oils from the foodstuff. If I added water, water and fat of course don't mix, so we wouldn't get much happening. And what I'm then going to do is I'm going to filter that into a clean test tube. Now I've already done that in preparation in true Blue Peter style. So this is the filtrate I obtained from another sample. And so I'm going to keep that there. I need to take another clean test tube and I need to half fill it with cold water from the tap. So we're going to let the tap run cold. That's fine. Just half fill that test tube. So this is now cold plain water. I'm going to add a small amount of the filtrate into the cold water. Don't tip it all in. You can probably see that a film has developed just there. That's called an emulsion. And what it actually is lots and lots of tiny fat globules or oil globules hanging in the water, floating in the water. That's a positive result for the presence of oils in my sample. The final test I'm going to carry out is to test the same food sample for the presence of protein. And this time, what I'm going to do, I'm going to take the third test tube from the original filtering process, and I'm going to add 5 or 6 drops of sodium hydroxide. You don't need to put your thumb over the end and shake it. In fact, I'll ask you not to do that. What I'm then going to do is I'm going to take copper sulfate solution, not the Benedict solution. And I'm now going to add the copper sulfate solution to the solution in the tube here. And what I'm looking for is the presence of a violet coloured ring. So just add it drop by drop. You might just be able to see a very thin ring at the bottom there. But for the most part, what we've got here is just the colour of the copper sulfate. So this is a very weak result, showing that there's a very small amount of protein in that sample of biscuit. If you look on the card that accompanies the practical, you'll find examples of much stronger reactions, and you're asked to fill in the table on your lab sheet to show what your estimate of the amount of protein is.